Assalamu alaikum girls. Today we will discuss about how our gas is collected. It depends on the gases properties. For example, there are some gases which are water soluble and there are some gases which are water insoluble but they are heavier or lighter. So if the gas is water insoluble, so we can collect it through water displacement method in which we use a gas jar. We need water in a tub and we need a delivery tube, and water will be displaced by the gas which is produced here. The next is, if the gas is water soluble, so obviously it will not be collected by water displacement method as it get dissolved in water. So they, at that time we have to check whether that gas is heavier or lighter than air. So if we know its properties that it is heavier than air, so we can collect it through downward delivery method. So just see there is no water, so the gas can be collected in this way, and if the gas is lighter than air, then it can be collected through upward delivery method as it moves up because it's lighter. And there is another way by which the gas can be collected. It's by using a gas syringe we can collect it. This diagram which is given over there about the collection of gas using a gas range. So for example, here a gas is produced for by any reaction, and then we attach delivery tube. We need a bung over there so that the gas won't escape into the atmosphere, and then it is connected through the this gas range. Now, if you get confused at how we collect the gas over water or how we collect the gas by the downward or the upward delivery method, so basically from this side, you have to attach it instead of this setup, right? If you are collecting it through upward delivery or downward delivery method, so from this side, you have to attach it with over here, right? Or over here. So this is the part of setup is given, so you connect it in this way, right, to collect the gas. So rest of the apparatus which I showed you in the previous diagram, you can attach it from this side, and then the gas can be collected in this way. We have a video of this one. A person is collecting a gas over water. You just see that a gas is produced in this test tube and it is connected with a rubber tubing, and then we have a measuring cylinder so that we can measure the volume of the gas produced itself. Now if we just see the setup, there are different setups which can be used. We are collecting a gas in a conical flask and we attach it with a rubber tubing. We can use this apparatus as well. Let's see how we set up the apparatus. We need a container in which we can keep water, right? And then we have, that person is just telling you that how to fill the test tube with water and then how you can keep it over there. But we are going to use a measuring cylinder, right? So measuring cylinder, you will fill that measuring cylinder with water, completely filled with water, and then you have to invert it over the container containing water, right? And invert it in that way so that water won't go out. Now just see, it's going to invert it. You can cover it, and then invert it in the container inside the water so that water won't come out, right? Now it contains water and a little bit amount of air is also there. But usually when we do it, we try that there should be no air in it. Only the gas which is produced, it will be collected. Then you can tie it by using this clamp and a stand. Right? There should be some space over there so that a gas which is produced and through a pipe, it can just move to this measuring cylinder. And you see, he has done the setup completely, right? And you have seen it in the start when the gas was produced, so how it was collected. You see? So in this way, water goes out into the container and the gas will be trapped in it. So this is the way how we collect the gas over water. Now the next is I have to show you the experiment about the chromatography in which we are going to find out about the leaves extract. So leaves contains different type of pigments and we have to just find out through chromatography. In this experiment, the pigments so in a leaf is what will be we need by throughout paper chromatography. So first of all, we cut the pieces of the leaves and then we use mortar and pestle to crush them so that a little amount of sand will be added so that the cells will be crushed easily and we get the extract. They'll add a little amount of alcohol as well just to have to get the extract in a proper way. A measured volume of alcohol will be added. Now they're going to crush so that the cell will be crushed and we get green pigment. And we are going to find out how many different components are there in this green pigment, right? Now they'll take a chromatography paper. This is the capillary tube. You may use a dropper and just put the green pigment over there 4 to 5 times. Then they dry it as well in between, but it's not compulsory that we dry it. I'm just trying to move it a little bit faster. Now they are going to have a chromatography paper. They'll attach it with a pin as we have done in the lab and they'll put it in a beaker. This is alcohol. Again, they are going to take alcohol and then they will put it in a beaker. Now we have to dip the paper into the beaker containing alcohol, but make sure that dot should not dip in the liquid. Liquid will travel up. You can see that the dye will be separated into its component. It's traveling up. You can see a little bit green color and little bit yellow color in it. It's a combination of two colors. Then just take out the paper and draw the line in pencil which shows you the solvent front, and after that you can calculate the RF values. Now to calculate the RF value, we have to measure the distances from the starting point till the green color or till the yellow color. In this way, we can find out the RF values. Right, so you can see the green and the yellow color. See that how we can measure the RF value. Is a yellow pigment, is a green pigment. We have to measure the distance between them, and then we can find out their RF values accordingly. As you know, the formula, it's the distance traveled by the spot divided by distance traveled by the solvent. So distance traveled by the spot will be from the baseline till the color, and the distance traveled by the solvent will be from the baseline till the solvent run. So this is how we do it. Alright, so that's all for today. I just want to show you the two things today. That's about the collection of gases over water and the chromatography.